Hello, everyone, and welcome to the RX Hope Table Talks, session number 4, More Than Just Gains or Losses, New Insights into the Functions of SCN2A. This session is being recorded. My name is Leah Myers, and I'm the Executive Director for the Family's SCN2A Foundation. We are very excited to be bringing this virtual educational experience to our global community. During these sessions, the families and caregivers of the patients with SCN2A related disorders will be referred to as the experts, and the presenters will be the professionals. The goal of the virtual table talks is to allow experts unprecedented access to professional researchers and clinicians working diligently to find a cure for our children. This is meant to be a casual environment, and your questions and comments are encouraged, as we are all here to learn from one another. To keep things running smoothly, please keep your audio on mute and use the reactions button to raise your hand if you'd like to talk. Alternately, you can send questions through the chat and we'll read them for you. If you haven't already, please take a second to introduce yourself and your SCN2A child in the chat. Perhaps their name, age, whether they have autism, epilepsy, or both, and where you live. I now have the pleasure to hand this over to Dr. Al George from the Feinberg School of Medicine at Northwestern University, who will be talking about the functional properties of SCN2A variants. Thanks for joining us, Al.
Thank you very much, Leah. And hopefully everyone can see my slides and hear me okay. It's a pleasure to be here, and it's always been really gratifying to work with your foundation and meet parents and their children and hear about their stories. And it is motivating to us, and it gives us context for the work we do. So I'm just going to give you a very brief, probably 10 minutes or so, introduction to what we do, and then just a little bit of a hint of the kind of findings that we are generating. The big picture is that SCN2A is the gene. It is coding for a sodium channel protein that's in the brain. It functions to let sodium into cells, and that triggers the electrical activity of the neuron, of the nerve cell in the brain. We can actually measure, yes.
Interrupt us for a second. We can't see your slides if you, they're not up.
Oh, well, I'm on my title slide, so I forgot to hit share.
There it is.
How's that?
Perfect, thank you.
Perfect, yeah. I haven't shown anything interesting yet, so you haven't missed anything. But I think I was just about to transition to say that because these are functional proteins, we can, in the laboratory, recreate them in cells that normally don't have sodium channels, and we can measure their properties. The whole process of measuring sodium channel function is a very esoteric science called electrophysiology. And I'm not going to try to teach you that today, but I just want to give you a hint of sort of what we do and what we learn. So first of all, I'm sure this next slide is familiar to everyone. This came from a 2018 review paper published in the Trends in Neuroscience. And this was the result of a workshop hosted by the Simons Foundation Autism Research Initiative. And this review article included all the attendees of the meeting. But Stephen Sanders really put this figure out there to try to illustrate at the time what we knew about the spectrum of diseases associated with SCN2A and possibly their underlying functional effect of the different genetic variations in SCN2A. And what this is to show us is that there is conditions of SCN2A fall along a wide spectrum. From one side of the spectrum on the left are children that have infantile epilepsy, sometimes leading to epileptic and developmental encephalopathy. There is a less severe form of the disease with also having infant onset seizures, but the kids so-called grow out of it and they don't have developmental consequences generally. And then on the far right, is the whole spectrum of autism, intellectual disability, with or without seizures, possibly having a later onset than the typical infantile onset seizures. The bar here in the middle, the color coding, is to try to illustrate the functional effect of genetic variation in SCN2A. When the variation is high, or when the variation gives rise to elevated sodium channel activity, we call that a gain of function, or GOF. And that was originally associated with the early onset epileptic encephalopathies. Whereas there was evidence from the autism literature that loss of function mutations, or LOF, was associated with that disorder. And so this spectrum of loss and gain of function being associated with either autism on the functional loss side or epilepsy on the gain of function side has kind of stuck in the field. And it is a useful paradigm, but I will tell you that our work so far, and I think the work of others, is beginning to realize that this is really not such a simple story. And so my point today is really to raise the question, are there really only two possible functional outcomes when there is an SCN2A genetic variant? So how do we determine that? Right now, we have an experimental workflow in our laboratory that takes advantage of a sequence of molecular and cellular and electrophysiological and data analytic steps. So we couple all of this together. We have a team of about 4 people. Some of them work exclusively to generate individual SCN2A variants in a recombinant SCN2A, meaning it's been, we've copied the DNA of SCN2A into a plasmid DNA, which can be replicated in bacteria, so we can grow a large quantity of it. We then take that DNA and we introduce it into cells that normally don't have the sodium channel, and therefore any sodium current that we see is likely due to the plasmid that we introduced. Then we use a form of electrophysiological measurement called automated patch clamp recording, and this allows us to make lots of measurements in parallel and gather lots of data in a shorter period of time than if we did it one cell at a time, which is the traditional way. And then we have built lots of systems to try to analyze the data that we generate in an efficient way. Now, this is a nice working system, but it's taken years to develop this and perfect it, and we still make tweaks to it. Most recently, we've kind of been learning about the type of cells that we use and their limitations. And we have a newer form, a newer cell type that we think is going to be better in the long run that we're now starting to implement that I think will actually be better for us in the long run. Now, what genetic variants in SCN2A do we study and where do they come from? Well, they come from a variety of sources. Here's just some of the sources that we deal with. We have certainly benefited over the years from the explosion of genetic testing that's done in epilepsy and other neurodevelopmental disorders. GeneDx, as an example of one commercial genetic testing company, has provided us with some data from their genetic testing services. They also deposit their data into the ClinVar database, which is a publicly accessible, freely accessible database hosted by the NIH that is a storage or warehouse of genetic variation data, usually provided by genetic testing companies. We've also gotten a lot of information from the literature, what has been published about SCN2A. We get a large amount of that from a database called the Human Gene Mutation Database, which curates the published literature and picks out genetic variation in any gene and catalogs it. So this has been a very useful resource. We've also gotten variants from the Safari projects, the more recent project they call Spark, and previous to that was just a lot of funded research projects. And then lastly, of course, our friends at families SCN2A and the ability to learn from attending conferences about what variants have been found in your children, and we've been trying to be very responsive to acting on that. This is just sort of the major sources of those variants. And then what we do is we work with a group led by Ingo Helbig and Ed Cooper, that's part of our Center Without Walls, to prioritize which variants make the most sense to study. Sometimes we are putting together sets of variants that are largely those associated with epilepsy, and other times we're really studying variants that are largely associated with autism. But as we're learning, there's a lot of overlap. Now, before we jump into the functional aspects, I just want to give everyone just a very quick physiology lesson. And this is basically the function of SCN2A. SCN2A is a sodium permeable protein, an ion channel. And most of the time, though, this channel is closed. It's not letting sodium into the cell. It takes some action on the part of the nerve to open the sodium channel. And that action is done by changing the voltage on the cell's membrane and push it to the more positive end of the voltage spectrum. That's what we call depolarizing the cell. When that happens, sodium channels immediately jump into action. They go from this sleepy, closed conformation, where they're not allowing sodium into the cell, to an active state, where now they're wide open and sodium is flowing into the cell as fast as it can go. That movement of sodium then triggers the event at the nerve level that we call the action potential, when that's the primary electrical impulse that travels along the nerve. Now, if sodium kept flowing into the cell for forever, the cell would probably bloat up and die. But that doesn't happen because the sodium channel has a very clever mechanism called inactivation, that soon after the channel is able to be opened, it closes again. And this is really something that happens very quickly, within 5/1000th of a second. So the amount of sodium that can come in is very limited, but it's enough to drive the action potential. But it's not too much that the cell drowns in sodium. The inactivation is a very delicate mechanism. And although it's shown here as just a little lid that closes, it's probably much more complicated than that, and a lot can go wrong with it. Now, once a channel is inactivated, it takes some time for it to recover and go back to the original closed state, so all this can start again. And we know a lot about the mechanisms of these transitions from state to state. But at the end of the day, when we record a sodium channel using the electrophysiological methods that we've learned over the years, this is the picture that we get. What you're seeing is a series of recordings from a cell where we stimulate the cell with varying degrees of depolarization, meaning we inject a little bit of electrical energy into the cell so that the membrane potential becomes a little more positive. And then we keep doing it and cycle this experiment over and over again until we've created this family of sodium currents that are all superimposed on each other. Some of them you can kind of see are sticking out. Others are all kind of piled up on one another. But this is sort of the profile of an activating sodium channel. When the sodium channel opens, sodium flows into the cell, and that gives rise to this downward deflection in our recording. But as the channel closes again in what we call the inactivated state, that measurement shows a falling phase. So we start out with 0, and then there's a big violent inward movement of sodium in this downward deflection, and then it all stops and it comes back to near zero, but not quite back to zero. And so we define several elements of this trace. For example, the maximum amount of current that's flowing in the cell we call the peak current. This entire distance from zero to the peak, we can measure in a certain way to give us what we call current density. And that's basically a measure of how much current, electrical current, is flowing in the entire cell. And then the little bit that's left over at the end here, we call the persistent sodium current. This is the little bit that just won't go away. Now, sometimes the persistent current is small, like it is shown here, and other times it's quite large. And this is a pathological current in most cases. Now, there's a lot of other things that we measure. I'm not going to go into those details, but I will summarize those in a later slide. But just to give you an idea of the diversity that we observe when we're studying different genetic variants of sodium channels, here's just a slide that shows 8 different variants. Now, on the left are variants that are found in healthy people. We call these population variants. For all I know, I could have one of these. Some of these are very common, some of them are less common, but they all pretty much, if I put the normal sodium channel next to it, they would all pretty much look the same. There's a little bit of variation in the height, but this is just some variability in the experiment. On the other hand, those variants, the 4 on the right side, are all pathogenic variants. These are disease-causing. The 2 on the top appear to have less current, well, somewhat similar current, but less functionality for a variety of reasons than the normal channel. It's most obvious with this one in the middle on the right side, where there's basically nothing. And then the 2 at the bottom, the current is spread out more than it is here. And these are channels that are not inactivating as fast. And some of them have even a little bit of persistent current. So these are what we would consider gain of function. So this is a loss of function. This one here is also a loss of function, but it has more subtle properties that tell us it's a loss of function. It's not obvious from this one experiment. And then the 2 at the bottom we would call gain of function, because over time, this slow or sluggish inactivation is allowing more sodium to flow into the cell over those few milliseconds. Now, we can summarize this kind of measurement, in this case, current density, across a wide range of mutations as illustrated here. Here's just some examples on the right. And what we've done is we've measured them, and then we've taken an average and we plotted it as this bar graph. And the height of the bar and the direction of the bar tells us a lot of things. So for example, if the current density we observe in a particular variant is the same as the normal sodium channel, then it would read 100%. There's a couple here that read right around 100%, or probably aren't that different from 100%. If it's a lot less than 100%, it's got a smaller current or a loss of function, we draw the bar to the left, color it red if it's got a high statistical significance, gray if it's not. And then if the current density is larger than normal, we position that bar going in the other direction and color it blue. And so here you can kind of see in this experiment, just looking at one property, current density. Yeah, there's many that are lower than normal and a couple that are higher than normal. So just based on this one property, we might say, well, here's some loss of function variance and here's some gain of function variance. But that's just one property. The other thing I was going to say is we've positioned the variants that we studied in this experiment to be either those associated with early onset epilepsy starting in the first month of life, or after the 7th month of life, those at the bottom here. And you can kind of already see that there's not, if we just look at this one property, there's not a good division between early and late. It seems like there's a lot of mixed. But again, I bring back, that's just one property. We look at several properties. Here are the major things we look at. Current density is one I've spent all my time talking about. I've mentioned persistent current. There are a lot of other fancy terms up here that we look at. And some of these terms are measured and they generate more than one numeric value. So voltage dependence of activation gives us two variables. Same with inactivation. Recovery gives us potentially 2, 3, or more. And so there's a lot of information that we gather. And for each of these individual properties, we can assess whether the difference between the variant and the normal SCN2A can be evaluated or classified as either loss or gain for every one of these properties. And then we can summarize it by using something like this, where we have in columns individual properties. So here's current density in the first column. And we have some other properties in the other columns. Here at the very end is the persistent current. And again, we can color each little box either blue for being gain of function for that property, or red, loss of function for that property. And then we can sort of start to see, if you look across the rows, which represent individual variants, what each variant looks like. Now there's some here, for example, this one down here, R1626Q. It's all either blue or no color at all. So this is a variant that we would easily label as an overall gain of function. M879T is one where it's predominantly blue, but there's one red one. And so this kind of gives us an idea of, this one's mostly gain of function, but now we come, whoops, sorry, I don't know what happened there. There's some variants at the bottom here that look like they're mostly loss of function, more red than blue. But for the most part, they're all pretty mixed. And some properties, which we think are more important for gain of function, like persistent current, sometimes is going to be the dominant property. And other times, it's just mixed in with other things. So it's very difficult from these experiments alone to basically tease out or select or cherry pick a couple of properties and say, you know, based on this measurement, we're going to call this a gain of function variant. Now that may be the right thing to do, and sometimes we may get by with it, but other times I'm not so sure. So I think I just want to leave you with this thought, which is the gain and loss of function binary classification strategy seems useful, seems a nice oversimplification, but I'm not sure it's right. And we need to go to another level of analysis probably to be able to really tease that out. So last thing I want to say is that I've been meeting with Ann Berg, who some of you may know is conducting the clinical trial readiness study in partnership with Families SCN2A. And she has gathered a large number of participants in that study. And along with the surveys and everything else comes information about specific genetic variants in SCN2A. And we've reviewed that recently, and we've looked at the list of what are the unique variants in that survey, and which of those variants have we already performed functional studies on, and which ones do we need to start working on. And so this image is of a model of SCN2A, the protein, and the position of individual variations that have already been studied are shown in red, and those that are yet to be determined are in blue. And or the ones that are function is known or should be known soon, some of these are in the process of being made and studied, And then the blue ones are the ones that we haven't tackled yet. And so we've decided that we would probably prioritize several of these blue ones to work on next. Once these data are collected, I think it might be helpful if we use the infrastructure that Anne has built for the CTRS to maybe be able to present the functionality of some of these variants in her database. And so we're starting some conversations about that. Lastly, I just want to thank people in my lab that have worked on this project and also our funding agencies. I've listed here 2 of the 3 people from Lurie Children's Hospital that have been helping us with this study. I failed to write in Anne Berg's name, but I mentioned her in person. So anyway, that's what I had to say. I knew it was probably a little longer than 10 minutes, but thank you all.
Thank you so much. That was perfect, Al. Thank you very, very much. We have a couple questions, but I was wondering maybe Dr. Helbig's talk might answer some of them, or we could ask them now. Which do you prefer?
I think we, because again, Ingo's talk may fill in the cracks of what I didn't talk about. I think we should probably wait for him to present.
Okay, sounds good.
If that's okay with everybody, can you see my screen and can you hear me well?
Perfect.
Yes, for both.
So first of all, hi again. I'm very happy to kind of present to you again. And so what we're going to talk about today is it's actually an extension from what kind of Al was just kind of talking about. I'm going to introduce a way how we can basically harmonize information about functional data in SCN2A. And this is a kind of new language that we're developing that we call Phoenix. And what I wanted to kind of show you, and I might want to skip over some of these slides if this takes too long, is that much of what our lab is doing, both in here at CHOP directly, plus in our kind of role as a variant curation team for the NINDS Center Without Walls, has to do with data harmonization and data curation. And it is, especially nowadays in medicine and more so in precision medicine, it is extremely, extremely important to understand what we talk about and that we have a common framework to kind of use data. Sometimes when I'm the second presenter, I do something relatively nasty. I kind of take screenshots of the other people presenting. So Al, this is your slide, yeah? And if you look at this is even, it has your pointer on here. I couldn't get you in a second when it didn't actually kind of move over this field. This looks quite complete. So this is something where we can say if data in all these variants is present in all this kind of information, we have this. This is so incredibly important. And where we typically kind of start is by saying, how can we make this happen? How can we kind of harmonize this data if this doesn't come from a single lab? But how can we do this if this comes from various labs? And this is one of these kind of slides that I would like to kind of show, is that data harmonization exists when it comes to genomic data. I think Stacy and I have given a talk about the G dots, C dots, and P dots, so the common language that we use for interpreting variant. We've given a presentation previously about how we harmonize phenotypic data, so our work with human phenotype ontology and data harmonization. And when we look at functional data, there isn't actually a common language. When we talk about peak current, we kind of, everybody knows what they're talking about, but the way that this is kind of reported in the literature and how this can be retrieved for scientific analysis across studies, across publications, sometimes across decades of data deposition, is often difficult. Yeah, and this is where we kind of come in with our harmonization effort. And I'm just going to skip over this quickly, but basically sometimes the way that functional data is reported in the literature can be different and can be often hard to disentangle if we don't have a common framework to kind of map this on. All the data is there. Electrophysiologists are kind of very detail oriented, but they sometimes use different forms and different formats on how this is being displayed. Here, just a quick kind of shout out to our data harmonization team. This is David Lewis Smith, who's one of my fellows in the UK, and he's recently kind of shown us how much work this is to actually harmonize data and kind of form these frameworks. This is for the human phenotype ontology. And we've started doing the same now for kind of functional data and for building a dictionary on how we can use this and kind of analyze this across the board, and we're going to use SCN2A as an example. So Phoenix stands for a functional electrophysiological nomenclature for ion channels. Yeah? We had to squeeze this in, and I think I got this idea of calling this Phoenix when I flew back from Chicago, and I was sitting at the airport waiting for my plane. Yeah? And we've been developing this kind of language or this kind of format almost over a 2-year process with an international team. So this was an ELTS team in Chicago. We had our kind of collaborators from Germany, from the UK. We've kind of liaised with ClinVar, with the official people working with the dictionaries that are used. Yeah? And this is a kind of screenshot of what this looks like. Yeah? So we can take all these terms that you may have heard about, a decrease in peak current, yeah? And kind of say, this is where this lives on our dictionary. This is where this lives on our ontology. And this is how these different components relate to each other. So we've kind of, in a way, hard-coded this in a framework that we can use any template or any term. And we know exactly what this means. We know that a severe decrease in peak current is a decrease in peak current. Yeah? This may be obvious to us when we see this data, but it might not be obvious if we do this across all ion channel results and would like to have computational reasoning for this. And why is this important? Why is this important for kind of SCN2A? It's because the data that is already out there accumulates quickly and is only getting more. Yeah? Right now, we know that there are 47 SCN2A variants that have been studied functionally. Yeah? And we can now kind of say, what do we actually find when we look at all these variants that were ever measured and ever reported in the literature? So here's just a quick overview of our previous study. Oops, sorry. Our previous study with HPO. So we kind of know that we can have functional terms, and we know that phenotypic terms allow us to kind of assess overall gain of function or loss of function. And here's what it looks like when we just kind of take all these terms that we have in the literature and kind of add them together. Those are literally hundreds, if not thousands, of different annotations that we've taken from the scientific literature and kind of harmonized in one format. Here is basically the corresponding kind of chart to what you've seen in ELTS talk previously. So those are variants that have been classified as gain of function or loss of function. And you can see that the frequency of these different kind of terms, whether it's a normal peak current or a decrease in peak current, this varies quite a bit between different variant classes. Yeah? So on the one hand, electrophysiologists like to really measure these kind of small parameters, but on the other hand, they often assign a kind of general assessment. They say it's a gain of function, it's a loss of function, or I don't know. You know? And this is how these variants kind of compare across these different categories. And let me just kind of quickly show you what happens when we just really drill into this and try to assess where all these different variants are. And so here is basically, let me just go back quickly. Here is one example on 25 gain of function variants. Yeah, we know that not a single biophysical abnormality, be it the increase in peak current, so the more sodium flowing through, or other activations on how channels kind of behave while they're being deactivated or depolarized. No single measurement is found in more than 40% of variants when we look at gain of function. So gain of function is what we call heterogeneous. Yeah? And when we take this further, is that when we look at these 114 individuals in the literature that kind of correspond to these 47 variants, we know that three main kind of parameters, three main abnormalities account for roughly 50% of what is going on. This is the kind of the right shift of the voltage dependence of inactivation, that the channels inactivate, and I shouldn't say slowly, I've learned not to kind of use the word slowly here, but with kind of increasing depolarization. They may be actually slower when they inactivate. They may have a persistent current, yeah, but factors like the peak current are less common. And Sridhar, who's kind of our data scientist, and this he has visualized it like this. So those are gain of function variance in SCN2A. And you can see this circle shows all these different kind of categories, all these different measurements that EL's lab and many other labs kind of measure for the SCN2A. And here's loss of function. Yeah, you can see loss of function looks quite limited. Yeah, there are only a few parameters that define loss of function, whereas the reported gain of function is quite heterogeneous. More than 80% of loss of function variance in SCN2A are loss of function to an absence or decrease in peak current, whereas gain of function is very heterogeneous. So let me kind of put this all together. When it comes to gain of function, we know that there's not a single biophysical abnormality that is found in one sort of all variants. This kind of accounts for all variants. Yeah? We know that there's a relatively homogeneity when it comes to loss of function. We know there's quite a heterogeneity when it comes to gain of function. And we know that 3 parameters mainly assess the kind of gain of function changes. And this is, we're not thinking about a homogeneous gain of function disease, but what we're trying to do is basically take gain of function apart and say, what is really happening with these channels? Can we describe this better? Can we describe this more precisely? So let me quickly maybe summarize this. And I was going a bit quicker because I want to leave some room for kind of questions. Existing functional data, all the data that's ever been kind of measured on SCN2A can be mapped and analyzed using a kind of common language that we've been developing in our center without walls. And this is something that computational scientists call machine learning ready. So we kind of transform existing data into a format that we can actually look at. This is important because those are really hundreds or thousands of annotations that are very difficult to kind of parse through manually. Yeah, we've already seen that gain of function in SCN2A, reported gain of function, is very heterogeneous. Yeah. We're currently looking across ion channels, across the sodium channels. How does this actually happen when we look at other members of the sodium channel family? And we're trying to integrate it with the clinical data. Yeah. And this is something that we're trying to basically open up right now to look at other features and try to make this more applicable. And here's just basically kind of a number of the kind of terms that we use. So we have like 4,500 annotations of functional data already. Yeah? You may say, why do you actually need this? It should be quite straightforward with a channel, a gain of function or loss of function. But if you really kind of try to map and analyze all these different components, this is a very large and very kind of complex data set. So let me maybe finish by giving you an idea of how we can link a new language with existing frameworks. So many of the clinical data or many of the genetic data on SCN2A lives in a database that is called ClinVar. So ClinVar is the official NIH database for variant submission. And ClinVar has accepted our Phoenix concept of this language as an official format for data deposition. So we're working right now on making this possible that we can actually deposit these terms that have real world meaning into a database that they can actually live in the data in parallel to the genomic data. Linked to this is that we're actually in the process right now and asking the question, well, the way that we assess SCN2A variants, can we do this better? And typically when you get reports back that tell you that a variant is a gain of a missense variant that is of uncertain significance or is likely pathogenic or pathogenic, this follows a certain framework, which is called the ACMG-AMP criteria. We're now officially tasked by NINDS to build a sodium channel variant curation group. And we have our kind of kickoff meeting tomorrow where we have a bunch of experts who kind of really work through these criteria and say, can we describe this better? This will include functional data, this will include phenotypic data, and a more precise way on how we can name and define ion channel function for every lab in the world that uses these criteria. And then finally, what we're trying to build is a very transparent and very clear data framework that allows us to capture data better. We can capture genomic data, we can capture clinical data, and now we're trying to kind of find a standard nomenclature or a standard language to describe ion channel function. I've given you the idea about the need for this controlled dictionary. So if we kind of move beyond a single lab, we need to combine information, harmonize data. Yeah, we've developed a language for this. And I've shown you as an example for SCN2A how we can derive knowledge for a disease by using these frameworks. I'd like to give a shout out in my lab to 3 people. This is Erin, who's now actually in medical school, who's left our lab, but is kind of will come back as a student hopefully soon. Yeah, and then Stacy and Shrida, who've been working on the Phoenix ontology and the dictionary with us, and will be kind of part of our SCN2A team going forward. Thank you.
Thank you so much, Dr. Helbig. That was amazing. I'm sure I speak for most of the parents when I say we just got used to gain and loss. So this is a lot, and it sounds super complicated, but I think this is going to help all of us to understand that we don't neatly fit in any boxes, which we already kind of knew. We do have some questions in the chat here. Christine, do you want to ask your questions directly? You might be on mute. Would you like me to ask them?
I can ask them. It's fine. I don't remember my first one though. Sorry. Has the loss of function and gain of function recently been reclassified based on the age of onset of the seizures? Because last year, my son was classified as gain of function, and now it's kind of shifted, but it also seems like he could possibly be a mix of function as well.
Yeah, I'm browsing the chat, and I think you say that your mother to Anthony, age 13. Is that right? Okay. Yes. Yeah. So he was diagnosed at age 12 months of age. When did his seizures begin?
Based on what I know now, probably around 10 months of age.
10 months. So that would be considered a later onset SCN2A epilepsy. It's certainly not infantile onset. And so, so his variant A1773T is a recurrent variant, meaning there more than your son have that variant. And based on just our functional studies, it is mixed functional variant, but I'm looking at my little drawing over here. Probably it's most, it's a very mixed property variant. I mean, I don't know how to say it any other way. It's not a clean loss or gain. So what that means, I think don't have that answer yet.
Yeah, and I think maybe just to kind of emphasize this, Ellen, Christine, this is kind of basically the topic of a presentation today. We have to think about more than gain of function, loss of function, if you want to do kind of justice to the complexity of SCN2A. Yeah. So gain of function, loss of function is the kind of the highest level where many variants kind of fall in, but there are variants that have a very mixed property, that are not easily classified as either or.
Okay, to ask my next question then, since we're on the same area, can that mix of function be based on the brain development at specific stages?
So a couple of things. When we study the variant in our very well-controlled experiment, we don't really, in general, take into account developmental stages of the brain, because we're not using the brain. We're using an artificial cell system so that we can isolate the sodium channel by itself and study its properties without the interference of the hundreds of other electrical signals that are in a brain cell. However, we do know that SCN2A undergoes a process during development that is called alternative splicing. And that simply means that the form of the protein is slightly different if you compare it early in development, probably starting before birth and lasting a little bit after birth. And it's a little different during that period than it is in childhood and in adulthood. We've studied that quite a bit. That seems to be a factor in how some genetic variants affect the channel function, but not others. And so I think in the case of a variant that has a strong effect early on, we've demonstrated that to be more significant when we use the fetal or neonatal form of SCN2A. But I think in general, what we're using is sort of the standard canonical or adult expressed form of the gene for most of our experiments. So anyway, your point is that the brain is developing. So the way I think of it, in the way you asked your question, made me think that I should say that the effect of the sodium channel variant and its functional consequence may differ at different stages in brain development. Maybe a variant with a mixed functional effect doesn't have much of an impact on an infant brain, but a 10-month-old brain is different. And the constellation of functional effects may have a much bigger impact on a 10-month-old brain. A lot is happening, a lot is changing, and the brain may reach a point during development where it's less tolerant to even subtle functional abnormalities.
Thank you very much. That was a very detailed answer. I appreciate it.
Ashley Taylor has a question, but she has a screaming toddler in the background. And so I'm going to ask for her. Her little guy, Hudson, started having seizures at 22 months. And his variant is typed in here. It's the one that starts with L1036W. Can you explain that variant and if it's been studied, and if according to 22 months age of onset, we're assuming it's loss of function. However, he's had a mixed reaction response to different sodium channel blockers.
Maybe I'll take this question because it actually relates back to Stacy's and my present, and Stacy's on the call as well, just to kind of help me out with some variant questions. So this is a frameshift variant. This variant actually does not need electrophysiological studies to be assessed on what it does. So this variant very likely leads to a complete loss of one copy of the SCN2A protein. And if L were to express this in a system, this would not even create any type of current because the copy or this protein wouldn't be even made. So when you see it where this protein has a frameshift, this is something that is a change that would very likely result in a disruption and basically a non-production of this protein. Yeah, I would need to look a bit more closely how close this is to the end, whether this may kind of escape some of these processes, but very likely when we think about frameshift variants, those are loss of function variants, and they can already be defined from the genetic data alone and often don't need functional studies. I don't know, Al, if you had any additional...
I totally agree with that. Basically, this so disrupts the protein's ability to be made properly that it won't work.
Yeah.
And we don't generally do functional studies on frameshift mutations. That's the little FS in the nomenclature, by the way, or what are called nonsense mutations, also referred as premature stop mutations. Because again, they so disrupt the protein's structure that there's no way it can work. The only exception to that would be if one had that type of mutation at the very, very end of the sodium channel. That might generate a functional sodium channel, although the few that we've studied, we haven't found one yet.
And I think the same would apply. I think that the same would apply to Renata's question, who also has posted about a variant that has a star to it. So that the star means the same, that this is basically a stop at position 243, and this variant plus the variant that Ashley was talking about, even though they have different positions, they would likely result in the very same changes on the level of the cell. Yeah? There is some variability how this presents and how patients react to medication. So even though the response to sodium channel blockers roughly aligns with gain of function, loss of function, there may be lots of variability between patients. So this is a larger rule that doesn't necessarily apply to each and every situation in a patient.
By the way, this kind of mutation, this nonsense mutation or like the frame shift mutations, were some of the SCN2A variants that were first found among the early genetic studies in autism. At the same time, there was a handful of mutations in SCN2A associated with epilepsy. Usually the more severe cases that were brought to medical attention early and were genetically tested on a more urgent basis. And so we had this extreme initial group of variants, those that were found in the autism, where they could point to them and say, well, these are definitely loss of function. Of course, in that discovery phase in autism, they found lots of mutations that they could classify, but they just kind of left them off to the side. In epilepsy, the most severe cases came to light first. Those early studies almost always showed mostly gain of function properties, but people were looking for that. They were assuming a gain of function would explain epilepsy. And so most of us were sort of looking for things that would be gain of function, and we just didn't kind of have that holistic view of things. So I think that explains how we got on this gain versus loss paradigm from the early data. And now I think over time, we've started to realize that's an oversimplification, and we should look for a more nuanced interpretation. Keep in mind also, everyone's genetic background is a little different. And that may be as big a factor in determining whether someone has epilepsy or autism based on their SCN2A variant. I'm not saying it's going to explain everything, but it's certainly going to influence what happens. And it may also influence how children respond to different drugs.
That's a great thing to keep in mind, the genetic background. And we do have another table talk later on. I believe it's next month, with Kevin Bender and Dr. Yang, who have a possible explanation as to why some loss of function patients develop seizures and others don't. So Ashley, I'll make sure that I get that invite out to you. These are great questions. Michelle, did you want to ask your question directly?
Sure, I can. So kind of a two-part question. With regards to functional analysis, number one, what criteria do you use to prioritize variants that you're studying? And two, do you plan on studying all SCN2A variants?
Yeah, so those are great questions. I think that I'll leave some room for Ingo to also weigh in. Our usual criteria for choosing variants is, number one, if it's a recurrent variant, occurs in more than one person, that increases our certainty that that is a disease-associated variant. We want to make sure it's not in the general population, so we search for evidence that it's absent in the population at large, and there are databases that help us answer that question. And then thirdly, we want to make sure when we study a variant that there is good information about the clinical features associated with that variant. It doesn't do us any good to study a variant for which we don't know whether it's epilepsy or autism or something in between. We want to make sure that there's really good clinical information about that variant. And if possible, that clinical information includes how they respond to commonly used anti-epilepsy drugs, such as sodium channel blocking drugs. And so those are things that we consider. Ingo is co-director of our variant prioritization and curation core of our Center Without Walls, and I'll let him give us his perspective on how they prioritize variants.
Yeah, so we've actually learned that the kind of selection of variants for these large-scale studies is a bit of a science in itself. I think one thing that Al was saying about the availability of clinical data is something that we've actually learned, also largely due to the fact that most variants we get from diagnostic labs and from big databases have very little clinical data. That's why we would always prioritize variants that come from sources like associated medical centers or databases that and Berg is kind of maintaining with the natural history study. Yeah. And then there's some additional considerations that have to do with kind of what the kind of goal of the study is. That is often initially we would like to kind of test variants where we are more certain that they are disease causing. So variants where we have to say we really want to kind of push the system to its extremes to say, can we actually reflect the very severe gain of functions and the loss of function, including some variants that we already know. Yeah. And then eventually what we're trying to aim for is what is called VOS resolution. And you may remember from some of the genetic talk, VOS stands for variant of uncertain significance. This is often a label that a diagnostic lab gives or attaches to a variant when there's very little information that we can use to understand if this variant is kind of truly disease causing or not. And this is often the case when we have variants in SCN2A that were only found in a single person, yeah, and where these variants may not be at the same position where other variants have been recorded, and we don't have complete data whether this variant is kind of new in the child. And we hope that functional studies, while they give us much of information about what a variant does, would help us kind of move variants into certain categories and understand how valuable these tools are for making SCN2A diagnoses more certain.
That's super helpful. Thank you. Chris, mom to Maddie, who has not had a seizure yet, or may never have a seizure, right, Chris? Would you like to ask your question about Maddie's variant? It looks like it's R119. Is that a I believe?
Oh, I see it. You're talking, yes, Chris Ray, R119I. And the question is?
Probably just hasn't been studied.
Just what does it do? Yeah, I don't know that we've looked at that one. That one doesn't ring a bell. And I would have to check to see if we've got that one on our list to do. 119 is not one that we've selected yet to make. So that one I'm sorry, I can't tell you much more about it. That's an L, huh?
That's okay, thank you very much.
It's an L, okay.
Or an L.
It's an L. Okay, yeah. Good to know.
I can maybe make the comment that I'm just using one of the tools right now that Dennis Lal has built. We kind of looked at variants across ion channels, yeah. And I'm gonna link this quickly, but we know this has not been studied in SCN2A, yeah? But a kind of corresponding variant has been seen in SCN1A and SCN5A, at least according to some of the databases. So this is a variant where we know some, sorry, I just pasted this in the wrong chat, where this variant has been seen in other situations. Let me just quickly share my screen. I want to show you what this looks like. Yeah. Here you can see SCN2A, position 119. This corresponds to 118 in SCN1A and 121 in SCN5A. And there's at least some annotation that a related variant at the kind of corresponding position in 1A and 5A has at least been reported in some individuals. And sometimes we can use this information to kind of draw some inferences on what this might do in the SCN2A channel. Yeah, and I think Dennis Lal, who might be presenting in this kind of series at some point, he'll be the person to kind of speak to this. And we've seen this over and over again, that these variants kind of may not be reported in a person, but a corresponding variant in a different gene has.
That's super interesting. Thank you. And you're right, Dennis is on our roster to do a table talk.
Yeah, and maybe Jenny, I would keep this question for him. This might be a good one. Yeah.
Okay.
I noticed a question from Jenny, and there was somebody else that wanted just, what is it? When I showed the heat map earlier with the blue and the red coloring of the squares, what does white mean? Well, white is neither blue nor red, but it really reflects the fact that we can't tell, for that functional property, we can't tell the mutant or the variant from the normal. Looks the same. So normal, white is normal. And so if you take a step back, a lot of these variants have lots of normal functional properties. They have a handful that are not normal. And they could be along a spectrum of loss or gain.
Looks like we have Michelle. Did you want to ask a question? Your hand's up.
Yeah, I have another question. This is from a family in Israel that is not able to get on because it's the middle of the night there. And they were just curious about their child's variant. It's E1211K. And if it had been studied.
Yes, I believe that's on my list here. I've just shared my screen. And I'll see if I can, well, I can't. I'm not in screen share mode, but 1, 2, 3, 4. It's the fourth from the bottom of this heat map. And you can kind of see it goes from pink to blue to red to white to white to white to blue to blue. So again, very mixed variant. And again, I would be cherry picking if I were, a particular functional property to say much more about it. But it has these sort of strange opposing effects on voltage dependence of activation, where it's given a gain, but voltage dependence of inactivation is a loss. And we see this a lot, where there is sort of this mixed effect on those two properties. And these are the hardest ones to sort out. That is going to need some advanced modeling or another kind of experimental system to figure out what does that mean, and how does that channel affect a nerve different from the wild type. That, I think we just can't predict those. But that's a very common scenario that we see.
In those mixed variants, actually, Dr. Helbig, how do you treat patients? Like when they come in and they have one of these mixed variants, how do you choose between a sodium channel blocker and a non-sodium channel blocker?
Yeah, so I should maybe kind of preface this by saying that this is often not a question that arises because we often don't know what the functional consequences are. Yeah, so I think that those are often decisions that physicians may need to kind of make way ahead of time. Yeah, I think typically in many children who have early onset epilepsy due to SCN1A changes, we would assume that there's potentially a gain of function component, and that the kind of risk of or that the benefits of sodium channel blockers would kind of weigh or would be stronger than the kind of risks of trying this. But I think this is always an individual decision where it really depends on what the presentation is, how old the child is, what medications have been used. Yeah? And typically we kind of get the functional information often later, yeah, where many medications already have been used. At this point, as I think from a purely clinical standpoint, I would be in a way cautious making too many or preventing medications into a certain direction based on the functional data, especially on missense variants, kind of maybe emphasizing what Al just said, is that we often don't know with this mixed variant what in which setting really kind of dominates this. But again, this is often a very kind of specific decision in a kind of at some point in time for each patient.
So Ingo, if you were seeing someone with a nonsense or a frame shift variant SCN2A, you might think about holding back on sodium channel blockers. Yes.
Yes, and this is why I emphasized missense variants, yeah, and especially missense variants where there's often little information. I think overall it has become quite clear over the last few years that kind of when it's a very clear frame shift or a very clear genetic loss of function variant, that we would be kind of very careful with sodium channel blockers. And I was kind of referring to missense variants where they may have some mixed effect. What Leah was just indicating, that where we would hope that the electrophysiology would kind of tilt us into any direction. And these mixed variants, they're just difficult. This is something where we're not at this point yet that we can make these statements. And this is where we need more data and more information for. But for the loss of function variants, where it's very clear genetically that this is a frame shift or a deletion or a splice variant, we would be, or many physicians will be cautious using sodium channel blocker.
The other scenario that, again, keeps being discussed is when a child has infantile onset seizures and an SCN2A missense variant is found. It seems more often than not, correct me, Ingo, but seems more often than not that those are the children who respond best to sodium channel blockers. I mean, there was a paper by Wolf et al. Years ago that made that point. Yeah. And again, anecdotally, it seems to be holding up. The exceptions, though, seem to be when the genetic diagnosis is made late. And now maybe a child is 2, 3, 4 years old, and it's finally discovered that there's an SCN2A variant. I'm not so sure that one would predict universally that there would be response at that point to a sodium channel blocker, because so much has happened. The brain is evolving, and there can be consequences of seizures. So it's just not clear to me that we know at what age can one see the most response to sodium channel blockers in a, quote, gain of function sodium channel mutation. It seems the earlier is better.
Yeah, and I think echoing this, I think early diagnosis is important, and especially for children with kind of infantile epilepsy. This is why we're kind of really trying to get testing done as soon as possible if they present in the neonatal care unit, or kind of if they present with infantile seizures. And in many situations, sodium channel blockers would be our first, would be amongst our first kind of medications to try. Yeah, and we've seen kind of good responses, but we've also seen responses that were kind of ambiguous, or may even be kind of lead to increased seizures. And I think what you may want to take away from the way that we've described the functional data is that early onset epilepsy in SCN2A, when it comes to functional consequences, what the specific molecular patterns are that result in this early onset epilepsy, which is kind of largely summarized as probably gain of function, can be quite different from child to child. Yeah? And there are some changes that are gain of function variants in SCN2A that cannot be kind of treated with SCN2A sodium channel blockers. We often don't know this initially. That's why we would always try. Yeah?
And over time, as we're gathering more data, this is why I emphasized before, when we're prioritizing variants for functional studies, we really would love to know precisely how the child responded to sodium channel blockers, because that is something that we can test in the lab and eventually create enough information that maybe we could decide whether or not there's a particular functional property that predicts response to sodium channel blockers. Right now, I don't think we know what that is.
Clinical trials are, being talked about a lot in our online support group, and as they come up, they seem to be aimed at either the loss of function or the gain of function patients. How are we going to know if we're eligible for these trials?
Yeah, I mean, Leah, you and I talked about this the other day, and I think that my sense is for the drug studies and the genetic therapies that are coming out for epilepsy with SCN2A, they're targeting what's being labeled as gain of function variants. I think it would be a mistake to exclude anybody unless they had an overt loss of function variant. That's my personal opinion, because we don't know. But what has to be done in parallel is every child enrolled in one of these studies for whom we don't know what the functional effect of the variant is, that should be high priority for doing function, because we will only learn how they respond and how that relates to the experimental function of the channel variant if we do them. So my hope is that there'll be that children won't be excluded because we don't know what the function is, because that's the case for most variants, but rather just exclude those for whom there is overt loss of function, frame shift mutations, nonsense mutations, big deletions, things like that. Yeah, those are not going to be gain of function. We know that without needing an experiment. Everyone else, perhaps, if the clinical picture would support the notion of a gain of function, then perhaps my opinion is they should be considered in the trial. I'm not spending the money to run the trial, so that would have to be somebody that decision would have to come from people who are involved. But that's my opinion. But in parallel, all those variants, every single one of them needs to be functionally studied and tested in the laboratory for the effect of the drug, so we can learn something.
That's good advice. Thank you. Michelle, did you want to ask your last question?
Sure. Besides ClinVar, is there anywhere online that families can look to see if there is information about their child's variant? And as a kind of a second question, will the functional data for SCN2A from the Center Without Walls, will that ever be made publicly available where families could look at it and see?
Yeah, so I think, Ingo has kind of set the stage for the first part of the question, I think. The second part is, indeed, we're planning to make all the data that we collect in the Center Without Walls publicly available in some form. We're actually writing up a paper now, but no one wants to wait for a publication to come out. That could take several months. We're trying to come up with a way, as Ingo has mentioned, to classify variants from the perspective of their functional effect and get that information into ClinVar. And ClinVar is open to everybody. Yeah. The other thought we had was to link it with our website. The Center Without Walls has a website. We have discussed in very general terms of how to build in a mechanism whereby someone could go and look up a variant, whether it's an SCN2A or KCNQ2 or any number of genes that we're actively studying, and to get that information well before it's been published. Now, that sounds great. Where the rubber meets the road is we need to pay our programmer to build a system that is not turnkey. It doesn't exist somewhere. It's not off the shelf, I should say. And second is, I tried to emphasize this in my short presentation, is this is a pretty dynamic process of doing these experiments and being certain that we have the right answer. And so there never will be a time when we run an experiment on a Friday, analyze the data over the weekend, and on Monday, we pop the data onto the website. We just don't want to take the risk of putting something out that's wrong and having that information used right away. So there will always be sort of a curing process of the data so that we make sure that it is right and that we're not putting out something that's not right. So we have to kind of come to terms with that. When that When that magic point is, I'm not sure, but we're learning, and we're getting closer to understanding that point for example, KCNQ2, where we've been able to study about 140 variants so far. SCN2A is much more complicated to work with, and therefore I don't think we will ever get to the point where we're 100% certain about anything, just because it's so complicated. But ultimately, we do want the information out there. And as Leah knows, we do share data on kind of a one-off when we can. And therefore, I'm happy to take calls or emails from people. I certainly will let you know where your child's variant is in the process. Whether we have a final answer or not will vary. So.
And maybe just to kind of add from my end, that actually when we look at SCN2A in general, we just kind of ask the question, what proportion of SCN2A variants have been studied, and what proportion can we make a kind of clear statement about, because there are clear loss of function variants, and we look at all the SCN2A literature, the answer is 1/3. Yeah, so if we were to kind of see a new patient with kind of an SCN2A disease, there's actually a 1 in 3 chance that we would have actually very good understanding already of what this variant is doing, either because it's a clear loss of function from the genetic part, it's a frame shift, or it's a truncating variant, or it's because one of these recurrent variants that have already been studied. The problem is are the novel variants. The problems are variants that have not been seen in other patients, and this is where we kind of slowly try to chug away and add the data that we have.
See, Carla put up a nice question, which is on average, if you don't mind, I'll read it. In average, how long does it take to functionally analyze one variant? From start to finish, and by the way, we don't do one at a time. We do batches. We're currently working on a batch of 36. And that process of making the variant at the DNA level, introducing it into cells, doing the functional study, repeating the functional study, so we're confident in what's going on, it takes a few months. If we were to drop everything we were doing and focus on one like uber important variant, we could probably compress all that into probably 3 to 4 weeks. And we were doing this for a while with our colleagues at Lurie Children's Hospital, where they were identifying, really early onset. They were identifying novel SCN2A variants with very early onset seizures. And we really tried to push things as fast as we could within a month to 6 weeks, including a full functional analysis repeated so that we knew it was right, plus a quick survey of response to anti-epilepsy drugs. And that was challenging, but we were able to do it. It just wasn't sustainable because we weren't really turning over very many variants at a time. So now we're trying to do more variants by doing them in these large batches, but there's lots of places where we get hung up and we have to push through, and yeah, it's challenging. But usually we try to sort of say quarterly in our lab, every 3 months or so, we want to put out a final product of a batch of variants. Now, it's not always SCN2A. It may be, that quarter, it might be a focus on SCN8A, and it's the same team doing that work. So I hope that helps.
It's very helpful. It's a lot of work. Does anybody have any other questions? You could feel free to unmute yourself and just ask if you'd like. Well, it looks like we might be ending 10 minutes early. I can't even tell you how grateful we are that you both took your time, and Ingo twice now to our families, to the experts here. It means so much to us that we have you on our side fighting for our kids every day. So I thank you, and I thank you on behalf of all of our families. As Al mentioned, he is starting to look at the variants that are in the clinical trial readiness study. Great opportunity to get involved in research and get your clinical data out there and matched up with your genotype. And as always, thank you for taking the time to join us tonight. It was great to see you all.
Thanks for organizing this, Leah. It was a pleasure.
Thank you.
Bye, have a good night.
Thanks all.